Family Psoroptidae. Family Psoroptidae. Species are highly specific to their host. Unlike Sarcoptidae, they cause a condition known as SCAB in animals like sheep, goat, cattle, buffalo, equine, cat, rabbit, etc. They do not burrow into skin. They are parasitic on surface layer, causing the formation of thick heavy scab rather than thickening of skin. The following species are valid: Psoroptidae ovesi, Psoroptidae equi horse, Psoroptidae nathalensis, Indian water buffalo and cattle, Psoroptidae carvinus, American sheep, Psoroptidae caniculi, rabbit, cosmopolitan, also occur in ear of goat, sheep, horse, donkey, mule, gazelle, etc. Psoroptidae morphology: body oval, no dorsal spine, no vertical city on dorsum of proposoma. Legs are longer and project beyond margin of body. Bell-shaped sucker on stall on the tarsi of or all the leg. The pedicle of tarsal sucker are long and composed of three segments in Psoroptidae. As each terminal, male has adrenal sucker, copulatory disc, posterior margin of abdominal or male prolonged into lobes. Prominent in Psoroptidae and Coreoptidae. Not prominent is Orthodactyls. Morphology: Psoroptidae ovis. Cause of sheep scab, an important disease. Australia eradicated. Britain 20 years back eradicated. It has now re-entered. USA still exists in certain states. Morphology: male. A pair of copulatory sucker in ventral side. A pair of large terminal tubercle bearing savory hair. Female. Possesses sucker with jointed pedicle on first, second and fourth pair of legs. Pubescent female also provided with a pair of posterior copulatory tubercles which are absent in ovigerous female. Ovigerous female will have a genital aperture on the anterior aspect of ventral side. Life cycle: egg laid on skin at the egg ovulation. Hatch into 3 days. Larvae feed for 2-3 days. Mold to nymphal stage last 12. Hatch is a state of leathery. Nymphal stage lasts 3-4 days before mold lethargic period 36 hours. Small nymphs will become adult. Pubescent female appear before male on the 5th day and the male on the 6th day copulation take place. Pubescent female mold to become ovigerous. Life cycle from egg to egg 12 day. Adult female life 30 to 40 day and each lay 90 to 100 eggs. Pathogenesis: punctured epidermis, suckling, serum exudates onto surface, coagulate and form crust. Wool get lost, fall out or pulled by sheep by biting and scratching. Bear crusty punch unsuitable for mite to multiply. They migrate to margin. Wool get matted in serum that oozes out. Treatment: dipping is best. Chlorinated hydrocarbons prohibited because of insecticide residue. Organophosphorus compound can be used. Malathion or Sumethion 50 EC. 12 ml of 50 EC MS is 1 liter H2O 1%. 20 ml of 50 EC MS in 2 liter H2O 0.5%. Zolon 35 EC 5 ml is 345 ml H2O 5%. 20 EC 5 ml is 195 ml H2O 5%. Deltamethrin synthetic pyrethroid butox 4 ml is 1 liter H2O 1% solution. Residual effect 15 day. Psoroptidae caniculi, ear canal of horse, donkey, rabbit, etc. Head shaking, ota, carry, cassage, ear canker. If not checked, heavy infestation to other part of body. Loss of balance occurs. Septic meningitis may ensure. Difference between Sarcoptes and Psoroptides. Difference between Sarcoptes and Psoroptides. Sarcoptes. Psoroptides. Sarcoptes cause scabby of a man and sarcoptic magn of sheep, goat, cattle, pig, equine, dog, fox, rabbit, and other animal. Notrades, notoedras cause notoedratic magna of cat, rabbit, and rat. Nem- Nemidocoptes affect fall. Sauroptes cause sauroptic magna of sheep, goat, cattle, equine, and horse. Autodect cause autodactic magna of dogs, fox, cat, and ferret. Coreoptes cause coreoptic magna of equine and cattle. Morphological feature: body globose, body oval, integument covered with scales and spine. Presence of 6 dorsal conical spine, no dorsal spine. Legs are short. Legs are longer and project beyond the margin of the body. Bell-shaped suckers, cranicular on stalk, pedicle on the tarsi of some or all leg. Same. Pedicel of the tarsal suckers are not segmented. Pedicel long and compound of 3 segments in Sauroptes, short and not segmented in Orodactyls and Coreoptes. Leg 3 and 4 of the female along and in bristle inside of sucker. Leg 3 of female only bear terminal bristle and not sucker, while leg 4 of both Sauroptes and Coreoptes bear sucker. Leg 4 of female of Orodactyls is feebly developed and without sucker. Leg 4 of all the male shorter than leg 3. Anus terminal, no. Adenal sucker copulatory disc in male with adenal sucker on the posterior end of abdomen. Posterior margin of the abdomen of male is not bilobed. Posterior margin of the abdomen of the male is produced into 2 lobes which are prominent in Sauroptes and Coreoptes but not so in Otodactyls. Coreoptes. Coreoptes, named after their host. C. Quae, fetlock of a horse. C. Capri goat. C. Caniculi, ear of a rabbit. C. Bovis. C. Quae ovis all belong to the species of Bovis. In horse, foot magna is leg on piston. Autodactyls, cynotes, ear of dog, cat, fox. Suborder Thrombidiformes. They have one pair of stigmata located on the gnatostom at the base of chelicerae. Some have no stigmata or trachea. Example: Dimodicidae, breathe through body surface, well developed pedipalps. Chelicerae adapted for perching. Subfamily Dimodicoideae. Family Dimodicidae. Host: dog, cattle, goat, sheep, swine, wild animal, rabbit, hamster, rat, mice, etc. Uncommon in cat and horse. Highly specialized skin parasite. Live in hair follicle and associated gland. Very host specific. Seen in hair follicle or dermal gland. Sebaceous gland, modified sebaceous gland. Example: meibomium, caudal, preputeal, and vulval gland. Also seen in modified sweat gland, some maxillary skin papillae and mixed sebaceous and sweat gland. Example: perianal gland. Morphology. Morphology: 0.25 mm, female, longer, elongated, cylindrical, body divided into head, thorax, and abdomen. Head has 3 jointed pedipalp, 1 pair chelicerae, single hypostome. Thorax, ventral side with 4 pair of stumpy legs. Each leg is three-jointed. Vulva in female on ventral side. On the dorsal side in male there is a longitudinal slit which is the genital opening. Abdomen transversely striated. Red mange of dogs. Red mange of dogs. Demodex canis. Red mange in dogs. In other animal disease caused is termed follicular mange or demodectis mange. Etiology: D. capri, D. ovis, D. phylloides, pigs, D. bovis, D. follicularum man. Life cycle: D. canis, complete in 3-4 weeks in dog. 3 nymphal stage. Effect on the host. Injury to host occurs when mite puncture with their stylet like chelicerae, chelicerae, the epithelial cell lining the hair follicle and gland to feed on the cell content. The host response in most cases lead to thrombosis of internal infestation. Pustules initially seen on abdomen, neck, leg, also feed face and thigh region. Dog have repulsive mossy odor or rancid odor. Itching is less in dog. Both forms are seen. Clinical sign evident in dog less than 1 year old, presumably immunodeficiency. First appear as mildly erythematous patch above eye and corner of mouth, associated typically with hair loss, then spread to four-leg trunk. Must resolve without treatment. Death occurs in severely infected animal. In generally predisposed or immunodepressed animal, and in case of secondary infection, it develops into chronically severe moist purulent dermatitis, pustular demodicosis, unpleasant odor, rancid or mosey. If generalized, there is intense redness, tenderness of skin which easily bleed. Death occur in heavily infested animal. Mild and moderate hypertrophy of the affected epithelia. In some cases, marked hypertrophy and cell destruction occurs. Opening of hair follicle and duct of gland are blocked. Dermal papillae formation and nodule, hair loss, secondary bacterial infection, inflammation, pruritus and pustule formation, staphylococcus. Lesion occur on face and head and then spread. If untreated, death due to emaciation and toxemia occurs. Clinical sign: two major clinical form. Scaly or sequamous demodicosis, papulonodular demodicosis, red mange of dog. Most common: dry scaly dermatitis, itching, thickening and wrinkling of skin, loss of hair, reddening of skin, sac infection, rupture of follicular cell, inflammation, purulent exudate, localized or generalized, repulsive mossy odor, red skin, bluish green, coppery reddish color, skin in tender and easily bleed. Papulonodular domodicosis, obstruction of hair follicle gland duct, lead to palpable cyst-like nodular swelling in the skin, trap mite within, common form in cattle, goat and pig. Lesion enlarge as the mite multiply along with cellular debris and glandular secretion. Eventually rupture lead to secondary infection and abscesses. Can rupture internally introduce mite into lymphatics. Cat follicular mite. Cat follicular mite, Demodex cati. Etiology: D. or D. gattoi, sequamous form associated with underlying immunosuppressive disease. Demodex bovis. Condition: papulonodular form. Population of 100 to 1000 mite per follicle. Result in dilated follicle, lead to dermal papules or cysts. As they enlarge, they can be felt beneath skin. Lesion concentrate in anterior part of cow, notably neck, shoulder, axillary region, udder, papular cyst enlarged to form granulomatous nodule when follicular opening get blocked by mite, keratin and other debris. Usually seen in cow treated by pregnancy or lactation. Dilated nodules vary in size from a pinhead to a chicken egg. Larger nodule rupture to produce suppurative source. The pus-like exudate consistency of toothpaste when examined will reveal the pressure of large number of deep voids. Economic loss, skin damage, defect in leather, tanning industry, loss due to diminished quality of processed cowhide. D. Tori, another species recorded in Gelosvolkia. Goat follicular mite. Goat follicular mite. Demodex capri. Dermal papillae and nodule nodes common in young animal, pregnant and dairy goat. Papules on face, neck, axillary region, udder. This later enlarged to become a nodule suppurate. Might are transferred via exudate. Nodular rupturing internally result in granulomas when in phagocytic cell try to destroy mite.